Hello and welcome to BIOS Builders. We are thrilled to welcome Sekar Kathiresan, co-founder and CEO of Verve Therapeutics, to the show today. Thank you once again for joining us.
Wonderful to be here, Drew. Thank you for the opportunity.
Yeah, thank you again, Sekar, for your time. Would love to kick things off here just with a quick, brief personal intro. Would love to hear more on your background.
So my name is Sekar Kathiresan, co-founder and CEO of Verve Therapeutics. Prior to these roles, I was a professor of medicine at Harvard Medical School and ran a research lab that studied the genetic basis for heart attack and high cholesterol levels. In addition, I practiced cardiology at Massachusetts General Hospital and ran the preventive cardiology section there prior to the current roles.
Absolutely wonderful. Quick and brief background sake, really appreciate it for the audience members who may not have seen your wonderful content prior to this episode. And before we dive in as well, if you could quickly provide a brief intro to Verve Therapeutics for us, would love to hear that story or that conversation from your perspective.
Yeah, so Verve is developing gene editing medicines to treat heart disease, specifically heart attack. And so think one-time treatment, permanent lowering of blood cholesterol. That's really what we're trying to do. Heart attack is the leading cause of death worldwide. Heart attack is caused by cholesterol clogging the heart arteries. And a key part of the solution is to get somebody's cholesterol as low as possible for as long as possible. Right now, that is being attempted with daily pills or intermittent injections, but relatively poorly so. The majority of patients with that approach of chronic care don't get their cholesterols to the right levels for long enough. And what we're trying to do is change all that and really convert or transform that model of care from chronic care, daily pills, intermittent injections, to hopefully a one-time procedure, a one and done, where after a one-time treatment, a patient can expect to have their LDL cholesterol, blood cholesterol low, and keep it low for hopefully a lifetime. So that's really the vision behind Verve.
Incredibly exciting and honestly concise explanation of Verve. We're incredibly excited to dive into the origin story and the future projections and beyond. So once again, Sekar, thank you again for jumping in. While we're diving in deep in here, I really just wanted to start with the origin story from your perspective, Sekar. In 2018, you helped co-found Verve, seeking to find that solution, as you mentioned, for the global epidemic that is cardiovascular disease. Really just taking a step back, could you tell us more about the genesis of Verve? What was the spark that started it all?
The spark actually was a grant competition from the American Heart Association. So in 2016, the American Heart Association asked people to put forward their best ideas to cure heart attack, cure coronary heart disease. And Drew, believe it or not, they're willing to give $75 million to one person to be able to prosecute that idea. The competition was actually called One Brave Idea. And the idea we put forward has actually become Verve. The idea we put forward was a one-time therapy, a gene editing therapy that would lead to deep and durable lowering of blood cholesterol. Why did we put forward this idea as our one brave idea to cure heart disease? The reason is, I spent about 20 years studying heart disease genetics and trying to understand why some people are at increased risk for heart attack, particularly heart attack at an early age, and why some people are protected from heart attack. And specifically, we were looking at the DNA sequence and asking what in somebody's DNA could be putting people at risk or protecting them. And all of that research over 2 decades led to one simple research insight, which is that if one's cholesterol is really low, lifelong, it's very hard to get a heart attack. And we got that insight from people who naturally had their cholesterol very low lifelong. These people had LDL cholesterols of 30, 40, 50 milligram deciliter lifelong because they had a gene naturally turned off in their body. And so, and because this a gene was turned off, they had the low cholesterol, they were protected from heart attack, and there was nothing else wrong with them. They had no other health issues. This really suggested that there were certain genes that were spare parts, that you could turn them off and all you get is health. So this led to this idea of developing a medicine that would mimic this natural situation where a given gene is turned off. And that's really how Verve, the idea for Verve came together. We put in this idea into the American Heart Association competition. Now, we didn't end up winning that competition, but we decided to go ahead and just do it. And over the next couple of years, from 2016 to 2018, we got some other people excited, particularly a venture capital company called Google Ventures, GV, and a few other VCs. In addition, we were able to secure some funding from them, as well as get the initial people involved in the company, and got the intellectual property necessary to be able to develop a CRISPR gene editing medicine for cholesterol. And so with all that in place over a couple of years, the team met weekly over about 2 years from 2016 to 2018. And then in 2018, we had raised $60 million and started the company.
It's an incredibly exciting genesis, in particular, paired with the competition that really spurred that innovation. And for yourself and the team's sake to make that leap, it's incredibly exciting, especially, and excited to jump into that genesis story, chatting through GV and those initial fundraising rounds. But really first, before we dive into Verve's approach, especially on the scientific side, I would really love to add a little bit more context for our audience here. As you previously mentioned, you discussed some of the current treatments that are in the CVD space. As we're talking about pills, other day-to-day treatments that might be needed to lower cholesterol. But really, I'd be curious from your perspective, Sek, and maybe this really does tie into the beginning of the competition, talking about the genesis of Verve in general. What really are some of those key unmet needs in the CVD treatment space? What do you yourself personally see as those limiting factors that are really the unmet sides for folks that are currently dealing with these serious chronic illnesses?
Yeah, it's a great question. So there are about 50 million people who suffer from atherosclerotic cardiovascular disease, or it's often abbreviated as ASCVD. This is basically the umbrella term for heart attack and stroke caused by cholesterol-driven blockages in arteries. Those 50 million people all need their cholesterol as low as possible for as long as possible to make sure they don't develop additional problems. Okay, how is that being accomplished today? Are attempting, how is ASCVD treated today? Well, it's treated with daily pills, intermittent injections to lower cholesterol. The challenge is that those medicines do lower cholesterol anywhere from 40 to 60% after administration. But if you look a year out after somebody gets started on their cholesterol lowering medication, more than half won't be on them at all, won't be taking them at all. So their cholesterol lowering is not 40%, 50%, 60%, but more like zero because they're not on any medication to lower cholesterol. And as a couple of additional statistics that will kind of bear this out, if you look at patients with a genetic form of high cholesterol called familial hypercholesterolemia, very few, less than 5% of those patients are actually at their LDL goal. if you look at all patients with heart disease, heart attack in the United States, less than half are on any cholesterol lowering medication at all. So these statistics kind of bear out the unmet need. The challenge of chronic care, the idea that somebody has to take a daily pill for their whole life, that's very hard because of issues of access, healthcare infrastructure needs, patient adherence. All these things have to come together to maintain, daily pills or intermittent injections. And so there is, I think, potentially a new approach that we want to try to bring to patients as an option, which is rather than the chronic care model, why not a one-time therapy that would durably and dramatically lower LDL? So that's really how we're looking to solve this unmet need that exists right now. Basically, replace the chronic care model, or at least another way to say it would be add an additional option to what's already available and provide patients with an option of a one-time procedure for lifelong LDL care.
Well, I mean, thank you again, Sek, as someone who's been an expert in the field for decades on the space and now leading the helm at Verve. It's incredibly exciting to see your perspective on the changes in the chronic care model and hopefully how Verve is disrupting the space over time. It's incredibly exciting. And as we're discussing your approach and really just diving further into the science, the research of Verve itself, Sek. So, Sek, we recently had the pleasure of hosting David Liu to discuss the future of genetic medicines from the academic and research perspective. For yourself, both as an academic and now as an executive leading in the space, I would love to gain some background from your perspective. Could you briefly explain base editing and potentially the benefits as opposed to prime editing or others compared therapeutics more broadly approaching the cardiovascular space? From your perspective, as you've seen the space evolved, both as an academic and entrepreneur, how the space was created, the underlying process, where we are now, maybe the potential of the technology as we're discussing the future of this single curative therapy.
Yeah, no, happy to do so. Maybe I'll take a step back and kind of just walk you through how Verve approaches the problem and the technology flexible approach that we've taken. So, we have a singular mission to really protect the world from heart disease. And the deep conviction that cholesterol, getting it as low as possible for as long as possible, is a key answer. So from there, the next item is, all right, what genes do we want to target to be able to get the cholesterol low? And these genes emerged from human genetics research that we and others did. And the genes are, PCSK9, another gene called ANGPTL3, a third gene called LPA. We have very good evidence in humans that these genes can be turned off safely, and the people who have the genes turned off naturally have very low cholesterol levels lifelong, are protected from heart attack, and are otherwise healthy. Now, that's one source of the targets. Another requirement that we use for which targets to go after are there are other medicines that have been developed against these targets and have already been proven to work in patients and safely do so. So that's so-called pharmacologic validation. So we require both of those things for us to be able to pick a target to edit and turn off permanently. And now we have the genetic and pharmacological validation for these three targets that I mentioned. The next piece of the puzzle is, okay, you have picked the targets. How do we turn the gene off? What gene editing technology will we use? In 2018, when we started, we had the targets and we picked two gene editing technologies to evaluate. One is so-called standard CRISPR-Cas9 or genetic scissors. Another was a new kid on the block called base editing, so-called pencil and eraser. And so we had both of these, and we got access to both of them. And what we've done over the last few years is compared both for their ability to turn off PCSK9 gene in cells, in mice, and in non-human primates. And after that systematic comparison of the two approaches in those three model systems, we've selected base editing as our preferred approach for our first couple of products. Now, let me explain the difference between Cas9, standard Cas9, and base editing. Standard CRISPR-Cas9, again, the analogy is genetic scissors, is that it's a protein that will go to a specific place in the genome that you direct it to using a postal address that is really provided by a guide RNA. And the complex will go to the specific place directed to by the guide RNA. And when it gets there, the Cas9 enzyme will cut the DNA, both strands, creating double strand breaks. Now, cutting the DNA is an effective way to turn off a gene. In contrast, what base editing does is it takes the CRISPR-Cas9 enzyme, and what David Liu did was disable the cutting feature and replace that cutting feature with an enzyme that would convert one letter, one DNA letter to another. So, for example, an A to a G in the case of adenine base editors. So now we have a protein that can still use the GPS localization feature of Cas9 when given a guide, but instead of cutting when it gets to that place in the genome that you direct it to, it actually makes a single spelling change in that place in the genome. So for us, for our first product, our guide RNA directs the base editor to the PCSK9 gene in between exon 1 and intron 1, where there's a specific A is changed to a G at one spot. And that spelling change that's made basically disrupts a splice site and turns off the gene. So this concept, this idea of base editing, again, David Liu invented, we got access to it, exclusive access to base editors for the PCSK9 target and a couple of other targets when we started the company in 2018. And what we've done is develop the medicine from there onwards. So we basically made refinements to the editor. We've figured out what the right place to edit is. We've developed the guide RNA. And importantly, we figured out a way to deliver the gene editing tools to the liver in the body. And that's done using a delivery system called lipid nanoparticles. The two pieces of editing tools, the guide RNA and the editor, are delivered as RNA molecules. They're packaged in a lipid nanoparticle, a little fat bubble, and that's what's infused into the blood. And that's how ultimately the editor makes its way into liver cells and eventually makes the spelling change that I mentioned earlier. So that's a brief description of standard CRISPR-Cas9 contrasted with base editing.
Thank you again for the background's sake and from your own perspective, diving in deeper to the delivery mechanisms through LNPs. It's wonderful to see from your perspective as well, and really just discussing more in detail about Verve's solution and through target selection and that focus. Really, just from that perspective, beyond the initial fundraising cycle you mentioned, Verve has just seen tremendous growth since your conception in 2018, reaching the clinic and getting closer, hopefully, to transforming care for ASCVD permanently. But with really taking a step back here, sake, with every genetic medicine company comes questions of scale and development, which Verve's really attacked head on from our perspective. Looking at the space altogether, what about the unique challenges that genetic medicines face, and discovery or development are you seeing today? And really, particularly when it comes to prediction of protein structure, function, off-target effects. Mostly we discuss kind of the clinical entrance here, but I'd really love to see from your perspective, sake, some of these challenges within genetic medicines that you were currently or previously alluding to.
Yeah. So, as I said, we started the company in 2018. At that time, we just had some ideas on pieces of paper, and we've kind of transformed those ideas to a product now that we have shown can work in patients. So when we started, what were the challenges? What do we anticipate to be the key challenges? And I would say there are broadly like four buckets of challenges that we knew we'd have to face. One is biology. So what I mean by that is, PCSK9, turning it off, what would the consequences be? And is it going to be effective? Is it safe? Now, we had a pretty good sense that that would be maybe the least of our challenges because of these other medicines that I mentioned earlier that have been developed that target PCSK9. There's a monoclonal antibody, there is an siRNA, and those medicines have been tested in hundreds of thousands of people in clinical trials and in clinical use. And so we knew that I think this challenge, this biology challenge, probably could be overcome, but it was still out there for our mechanism of action. The second major area would be technology. So could we actually use this new technology, gene editing? Would it work in patients? Nobody had previously edited a base edited a gene in the liver. So that's a technology challenge. The third is regulatory. What we're trying to do in vivo gene editing, so-called in the body gene editing, there was no regulatory framework for what would be needed in terms of preclinical studies to be able to take this new approach to patients. Then lastly is a very general challenge is commercial. We may be able to make the medicine, get it to work effectively in animal models, get it to work effectively in patients, satisfy regulators, get approval. Then the question is, okay, would patients want it? Would physicians prescribe it? And will payers pay for it? So that's the whole kind of commercial question. Of course, we still have a few years to go to tackle that last one. But these are the four major buckets of challenges that we anticipated when we started the company. And what's been satisfying to date is we've been able to address pretty firmly the first two, the biology and the technology items. We're starting to made headway on the regulatory, and to the point where we have accumulated data on, some patients already and are excited about the path ahead as we move from the current phase 1 trials to ultimately, hopefully, an approval in a few years.
Of course, sake. And thank you again for the breakdown from development to commercialization, the thoughts and challenges of really being a pioneer in the space moving forward in genetic medicines. It's certainly an exciting time in field as a whole, but hearing your perspective as well of how Verve is breaking it down and how you as a executive in this is really leading the charge a lot, both in regulation and commercialization. So thank you again for breaking that down. And we hope at some point to bring in and potentially have a follow-up episode discussing the look back of that process. I certainly know it's going to be an exciting few years in genetic medicine. To continue the story as well, as we're discussing these challenges of scales, and really as we're looking forward as the next steps in commercialization for genetic medicine. Really, in this past June, Verve recently entered a global collaboration partnership with Lilly to advance Verve's in vivo gene editing program targeting LPA. really just to further this conversation around scale and this issue around commercialization. Potentially, could you take a moment to describe the opportunity of this partnership and what this means to Verve?
Yeah, so we've in 2023, we've developed a relationship with Eli Lilly. And what we're looking to tackle is literally the world's leading cause of death. A very um, a global problem, a very big problem. And we have an innovative solution, we think, to that problem. And but it's going to take, I think, a fair amount of resources to get this to the finish line and ultimately get it to patients. And it was clear to us that we'll need a partner along the way. And to date, our partner, as of last year, was Beam Therapeutics. And Beam is the company that we had licensed the base editing technology from. And in return for that exclusive license, they held some minority rights for our PCSK9 program and the second target, ANGPTL3 program as well. What Eli Lilly did this past year was purchase the rights that Beam previously had. And so Eli Lilly is now our partner going forward rather than Beam. And we welcome this development because Lilly has a considerable expertise and experience in the cardiometabolic space. They have the resources necessary to ultimately see this kind of development through all the way to registration and commercialization. And they really have developed conviction in this approach of one-time procedure for cardiovascular disease. And their general philosophy has been pick gene targets of high conviction and then offer multiple treatment modalities against that specific target. So, for example, for the same target, you might have a pill, you might have an injection, and you might have ultimately a one and done option as well. And so we are excited to work with them on actually a couple of different targets to develop the one and done component. And right now, as we're in the phase one, their involvement for PCSK9 and ANGPTL3 is relatively small. But when we complete the phase one, at that point, they're going to be able to look at the data and then opt in to partner with us for future development, phase two and onward. And when they opt in, they will end up picking up a portion of the expenses of development and commercialization in the United States. And in return, they will get about 50% of US profits. The rest of the US profits are, the other 50% is Verve's. And then importantly, all of ex-US is Verve, 100% Verve. And Verve controls clinical development and books revenues. So that's really the nature of the partnership. But we're super excited to bring them on board for this journey to develop gene editing medicines for cardiovascular disease.
Thank you again, Sek, for sharing the behind the scenes beyond the announcement and your own perspective. It's certainly an exciting time for it. It certainly poses the question as well, potentially, if you could share more about the future collaborations Verve is excited to partner with or organize as well beyond Lilly. Would love to see, as you're looking on these next stages, what you're excited about or what you're seeing as we mentioned. This field and commercialization certainly takes a village. Curious to see from your perspective on other collaborations, other partnerships that you might be excited to begin to allude to once we're going towards the future here.
Yeah, I think that, for our first few targets, PCSK9, ANGPTL3, and LPA. So these are the three cholesterol targets. Lilly is our partner. And so we see an exciting future with them as we, again, try to tackle the leading cause of death in the world with these products. We've also entered into a partnership with Vertex Pharmaceuticals for a liver disease target. We have not disclosed that target yet to the public, but it's an exciting collaboration because we get to work with Vertex, a terrific company, extremely innovative. We are using a new editing approach for that target. So again, speaks to that technology flexible philosophy that I mentioned earlier. We're not wedded to one editing tool or another. Rather, we look at a target. In this case, this is a liver disease target, and we say, what edit do we need to make? And what might be the best tool to make that edit? And then we go with that. So in this case, we're developing a custom editor bespoke for that target. And so with this partnership with Vertex, we're basically able to, refine and develop our editor development capabilities. And so that's an exciting area. And that partnership is now almost 2 years in and making very good progress to try to solve that problem. And so that's really a description of our partnerships to date.
Incredibly excited to see the process on both of these partnerships, and thank you again for diving into this further, Sek. And really appreciate both the broad perspective on both the genetic engineering space, but also the specifics of how Verve is tackling these challenges head on, just directly at the helm and the future of a leading company in the space itself. Just to take a moment, though, I really want to take a step back here and really just discuss the major challenges and accomplishments Verve has faced from your perspective, Sek. Really, Verve has found incredible success since its founding, pioneering a new marker for genetic medicines, acquiring several promising partnerships, including Vertex, like you mentioned, Lilly, among many great others, especially going public in 2021, raising nearly $270 million that ranked as one of the largest in biotech that year. Was certainly exciting. Sek, just looking back, what have been some of the greatest challenges you faced scaling at Verve from your perspective?
Yeah, I think I mentioned the kind of general categories of challenges earlier, but I think the biggest challenge we face to date is regulatory. And so, what we're trying to do really, it's molecular surgery. That's the term I like to use. That's probably the best analogy in the sense that this is not less a drug and more a one-time, molecular surgical procedure and intended for lifelong benefit. And so what's the regulatory framework for thinking something like this through in terms of assessing risk benefit, even to the point of, okay, it's okay now to treat a first human with this, because there's no precedents. There were very few precedents, I should say, or before we got started for this kind of in vivo liver gene editing, particularly in the United States. So we've had to kind of pave the way. Now we were able to evaluate the risk benefit in preclinical models in terms of cells, mice, and non-human primates. We also evaluated the risks, particularly of so-called off-target editing. So this is the idea that, hey, I'm looking to make a single spelling change in one spot in the genome to turn the gene off, the disease-causing gene off. But might I make changes in it somewhere else? Could there be a change somewhere else that could be harmful? so-called off-target editing. So we've evaluated that extensively. And what we find for our product is actually we get the on-target editing, but really no evidence of off-target editing in the liver, for example. So that a really high level of specificity. The changes being made at the one spot, A to G, but nowhere else. So all of that data we put together and submitted applications for our get permission basically to treat a first human being with this investigational medicine. We did that globally. We did that in 3 countries, New Zealand, UK, and the US. Our initial clearances came in New Zealand and UK, and that's where the first 10 patients were treated. In the US, our initial application was met with additional questions. And so it took us another 9-10 months to answer those questions with additional experiments and additional data. And ultimately, as I mentioned earlier, in October of 23, we got clearance in the United States, and we're in the process of activating US sites to be able to treat a first patient in the United States. But that regulatory interactions, sets of interactions, probably have been the biggest challenges that we've faced to date. But thankfully, we've been able to kind of overcome that challenge and have a path forward now, not only ex-US, but also in the US to develop these in vivo gene editing methods.
It's incredibly exciting to see from your perspective, Sek, and thank you again for diving in from your perspective on the regulatory issue, especially leading the helm on the frontier of this space. Certainly covers a lot of ground for hopefully many great founders moving forward, but it's wonderful to hear from your perspective. Really on that same note, we've mentioned some of the great successes that Verve has had just on our episode today. Just really from your perspective, your own side here, Sek, on this same note, as we're discussing challenges, what have been some of the greatest accomplishments or milestones that you feel that you've reached or you'd be proud to share, at least in our episode today, at the moment, from your perspective?
The biggest accomplishment is the medicine works. We started in 18 with an idea, and an amazing amount of teamwork. We now have about 260 people in the company. Those people over the last 5 years have worked incredibly hard to get to the point where last year we showed that, yeah, you can infuse a drug into a human being. The editor makes its way into liver cells and ultimately to the nucleus where the DNA resides. The editor scans the genome, chromosome by chromosome, in a given cell, finds that one match to the guide, sits there in the PCSK9 gene in between exon 1 and intron 1. There's a single spelling change made. This is happening now in basically every liver cell in that person's body. The gene is turned off, and the blood cholesterol comes down 50 to 60% in a couple of weeks, and then has stayed down now for 6 months plus. So it's pretty remarkable. What really was almost science fiction, really, in 2016, 17 when we started, now is a reality. And Verve was the first company in the world to show that in vivo base editing can work in patients. So that's probably our biggest accomplishment to date.
Oh, of course, certainly. And I mean, the amazing accomplishments that Verve has had, the team formation, the funding, the technology itself, absolutely working. It's incredibly exciting. And thank you again for alluding as well to the incredible progress that both yourself and the team have made in the frontier of patients. And dosing those patients certainly must have been an amazing experience as well last year. So again, congrats to yourself and the team, but thank you as well. Really, Sek, I want to take a moment here to discuss towards the end of our episode here, really just the great beyond. Having discussed Verve and your approach to date, I'd really love to project out to the future here. Just starting off here, Sek, what's coming next for Verve from your perspective?
Well, I think we're going to be continuing to develop these medicines. This year, 101, targeting PCSK9, as I mentioned, will complete the Heart-1 trial. And then 102, the product that also targets PCSK9 with a different delivery vehicle, we'll be getting human data here this year for that 102 program product. Next year, we will be basically looking side by side at the two products, 101 versus 102, comparing the human data, and likely picking one of the two to take forward to later stages of development, the phase 2 and phase 3. So what I can see in a few years is those phase 2 and phase 3 studies, and ultimately, hopefully, at a point where we'll have an FDA-approved drug a doctor can write a prescription for, and ideally by the end of the decade, before 2030. That's for PCSK9. For ANGPTL3, a similar story in terms of phase 1, phase 2, phase 3. But the initial patient population here is called homozygous FH. This is a rare orphan disease. And so we expect that this medicine could get to market even quicker than the PCSK9 product because orphan diseases, rare diseases, can sometimes the development path can be quicker. Then, as I mentioned, we have the LPA product that we're working on with Lilly, and that's also going to be developed in a very similar way. So I think the next few years is really focus on taking these products forward. We also have a couple of other products in the pipeline that are even earlier. I mentioned the collaboration with Vertex. There's another atherosclerosis product as well, and we'll be moving those. I think the big questions for Verve are we going to be tackling other diseases beyond heart attack? For example, other organs or diseases in other organs. And I think those are things that we've been thinking about, working on in the background. And I think our focus on these very specific targets, these very specific products, has been probably the key ingredient for our success to date, the focus. But given our success to date, it does give us a little more bandwidth to think about, well, where else could we apply our energies and maybe have impact as well. So that's something that's ongoing within the company and part of our future.
And thank you again, Sek, for alluding to this discussion as well as someone that is really pushing forward several different assets in specific fields, but also looking at where Verve can approach next as well. It's certainly an exciting place, especially as someone that really truly is on the frontier. And I almost wanted to press you there in the moment as well. Just discuss that a little bit further from your perspective as you discuss the assets that you're currently pushing through in partnerships. Really with any type of platform company with a diverse list of assets, there's always almost a debate between vertical integration or horizontal platform expansion. From your perspective on the inside ball of Verve, how have yourself and the team really been thinking about these opportunities?
I think we've been very focused, as I said, on this disease, atherosclerotic cardiovascular disease, and these targets. Because it's the leading cause of death in the world. So if we have impact on this problem, that should be good enough for a single biotech. We shouldn't need to kind of go looking too broad, And if you think about our pipeline of PCSK9, ANGPTL3, and LPA, I think about the opportunity not just as those three rows on a pipeline, but also our potential for expansion, indication expansion for each of the products. So so-called like columns of expansion. So the first column for each of those three products is the genetic form of the disease. For PCSK9, that's a disease called heterozygous familial hypercholesterolemia. For ANGPTL3, that genetic disease is called homozygous familial hypercholesterolemia. And then LPA, it'll be people with the most extreme LPA levels. But from there, from that first indication, our ambition is to expand further. So for example, for a PCSK9 product, we'll go from heterozygous FH to atherosclerotic cardiovascular disease. So that's basically anybody who's had a heart attack. That's about 50 million people in US and Europe, 200 million people worldwide. So that's kind of like the second ring of the bullseye. It's kind of a bullseye model of clinical development. The inner ring is the genetic disease. The next ring is kind of atherosclerotic cardiovascular disease more broadly. The third ring, the outer ring, is prevention. So basically, why wait until somebody already has a heart attack to lower their cholesterol? Let's do it before so that you can avoid the first heart attack. So that's how we think about the pipeline. So you can see with this model of three products, three rows, but then three columns for each product, the opportunity is enormous. So we don't need to go much further to really have dramatic impact on health. That being said, we are thinking, as I said, about other organs, other places where we could take our technology, take our learnings, and apply. But that's in its early stages right now.
Certainly as well. And I mean, great to hear from your perspective, and completely agree on our own right. The issues, the targets that yourself and the team are focusing on Verve certainly are the exciting and grand problems that we are seeing in the healthcare and patient perspective in the current field. So thank you again, Sek. Before we really jump into our more quicker rapid fire questions, I really wanted to take a step back for a few quick questions before we wrap up here. Thinking about the patient impact and looking to the future with the adoption of Verve's platform and the scale up of the broader precision medicine space, we'll hopefully see the cost barriers changing for the genetic medicine space in discovery, development, delivery. From your perspective, Sek, as someone really leading the helm here, when do you really see this tipping point happening from your own perspective?
Well, actually, I think hopefully it'll be with products like ours. To date, advanced medicines like gene editing or gene therapy have been restricted to rare diseases and cancers. And the price points have been millions of dollars a dose. Now, that's not going to be us because we're not talking about a rare disease. We're literally talking about the most common cause of death in the world. Lots of patients. Second, the cost of goods here for us could give us a lot of flexibility on how we approach access. Our product, in 2018, we proposed this idea of editor carried in a lipid nanoparticle. That was pre-COVID. Then COVID comes along in 2020, and the COVID vaccine, which is mRNA packaged in a lipid nanoparticle, looks just like our drug. Now we know that kind of product, about 100 micrograms of it, can be made for about $3. Now, we're not going to be giving 100 micrograms. For us, our product, we give about 50 milligrams. But even if you multiply up, you're talking low thousands cost of goods. So that puts our setup in a very different territory than what you're seeing right now for gene therapies or gene editing medicines in terms of millions of dollars a dose. We think ultimately the reference points for us will be more like other one-time procedures in cardiovascular medicine. So think a stent procedure, a bypass surgery. Now, these are all one-time surgical procedures that are done. Millions of these are done a year in US and Europe and worldwide. And so that's probably the better analogy for how access will look like for our one and done gene editing medicines for cholesterol.
And I completely agree on that side, Sake, and I appreciate the alluding to really the mental shift that might have to focus on, really pivoting this to a one-time procedure and how that might have to change the public's perspective, but also regulatory perspective on how really these precision medicine companies are viewed or are delivered out to patients. Um, as you were mentioning, just talking about Verve's platform and potentially just the next steps on technology, other innovative approaches that are approaching the genetic space. Um, Really, from your perspective as a close-off question here, thinking more broadly, both as an academic and executive, are there any additional enabling precision medicine technologies you're excited for that will help Verve's platform reach its goals? Any initiatives, ideas, call to startups, things that you're focusing on in particular?
Well, I think the key there are several different challenges ahead, but one key one is delivery. We've been able to get things to the liver um, quite well. But if you want to get this editing machinery to other organs, there's still a lot of work to do. So that's probably the one area that I would kind of keep an eye out on. Where's the progress there being made? Which approach? And we're actively kind of surveying the field there.
Amazing. And great to hear from your perspective as well, Sake. Really, but before we come to a close, a few rapid fire questions I'd love just to cap off this episode here. Thinking a little bit more broadly, yet again, Sake, really as we're looking towards the future here and discussing the potential impact of where Verve could position itself, but also just looking at the genetics medicine space as a whole. Really, we love to ask as well, imagine that it's 2050. From your perspective, say, could you really describe the genetic medicine space? What challenges have we addressed? What diseases have we treated? And really, where would we be going from there?
Yeah, I think there's a tremendous promise for these editing medicines to treat a range of diseases, both rare and common. I think speaking kind of from my own experience as a cardiologist, what I'd like to see is a future where, heart disease, specifically heart attack, and the blockages that lead to heart attack, can really be eradicated, you know. And I think the way to get there is to get, as I mentioned earlier, get the cholesterol as low as possible for as long as possible. By 2050, I think that the care for heart attack could look like this. Somebody comes in, they have a chest pain, they have heart attack, they found to have a blockage in one of the arteries that led to the heart attack. That blockage is cleared with a stent procedure. And then shortly thereafter, they get another one-time procedure, our infusion, and their LDL comes down 60% and stays down for the next 30 years. That's the vision, I think, for future heart attack care. And I think in parallel, I'd like to see, further development of that prevention concept I mentioned, where we don't need to wait until somebody already has a heart attack to administer such a therapy. But somebody turns 50 or 40, they have a strong family history of heart attack, they have high cholesterol, and they get, for lack of a better term, a liver tune-up, where they have a one-time therapy that basically dramatically and durably lowers their LDL so that they never get their first heart attack. So those are the two directions that I think I'd like us to move in. And I think by 2050, hopefully there'll be a fair amount of progress toward those two goals.
Great to hear from your perspective, Sake. And want to pose this back to yourself and the company as well. Still 2050 in this, right? Where is Verve from your perspective?
I think Verve would, an independent company, large market cap, solving heart disease around the world. That's where I'd like Verve to be.
Amazing. Sake, as we're closing off the audience here and finishing up our conversation, any other closing thoughts or shameless plugs you'd like to share with our audience here?
No, I just closed coming back to kind of why we started this. We started this company because of a deep conviction that the answer to heart attack is getting cholesterol as low as possible for as long as possible. And gene editing offers a unique approach to accomplish that. And we set out, to really see if we could make this happen. And we're, I would say about halfway through that journey. It takes roughly 10 to 12 years to go from concept to a new medicine, particularly a new treatment modality like ours. And my greatest hope is by the end of this decade, a doctor will be able to write a prescription for one of our medicines, and that medicine will have impact on lots of patients. Thank you.
Cannot think of a better way to close out this episode and just a fantastic hour of this conversation's sake. Thank you once again for joining us. We're incredibly grateful for your time and hope to have you on for the recap and review of the amazing impact that Verve has had in the future. Thanks once again.
Thank you, Drew.